Hello, and welcome back to this video on chapter 3, talking about the pressure volume cycle uh, and some other things. So let's get into it. First off here, this will be the ventricle right here that we're representing. And we're looking at the pressure in the ventricle as well as the volume. And the pressure is always measured in millimeters of mercury. The volume is always measured in milliliters. Okay, so um, right here is where where the ventricle starts to fill up, right? So this will be during uh, rapid inflow, and also atrial systole is this little bit here. And this is just where, as you can see, the volume is increasing and the pressure is not really changing very much. So what this dot here then would represent is the opening of the AV valve. Then as that goes down, once it stops, right, then that will be the opening or the closing of the AV valve. Okay. Now, right here, then this will be isovolumetric contraction because the volume here is changing, but the pressure is increasing a lot. So that means the ventricle is contracting, the volume's not changing. And then you get up to this point, and then this is during ejection. And so this dot right here can represent the opening of the semilunar valve. And over here will be the closing of the semilunar valve. And again, when the valves close, it makes the sound, right? So we have dub there, and up here. Okay. Now there's a couple really cool things that we can learn from this graph. First off, if you can think about in this graph on average, how much pressure changes per amount of volume. So if we do change in pressure over the change in volume, you think about when the ventricle is completely contracted, right? So this ventricle here becomes completely contracted. That's a terrible word. It becomes completely contracted. It gets really small. When it's completely contracted, how easily would it be to move the ventricle there? How much pressure would you need inside in order to increase the volume of the ventricle? Now that idea, right, that's basically how hard the ventricle can contract. And so that's inotropy. Another word that we have for this is elastance. And so an increase in elastance means that there's an increase in inotropy, which means the heart contracts harder and it would be stiffer. So even though if you think of something as elastic, it means it can stretch more. In this case, elastance is not. Elastance is actually preventing it from being stretched when the ventricle is contracted. Now, on the graph, the way that we can kind of see inotropy is if we made a tangent line right here. So if we take the immediate slope at this point, then and we build a line off of it, then it would look like this red line. And really what it comes to is at zero pressure in the ventricle, right? And at zero pressure, there would still be some amount of blood in the ventricle. But at zero pressure, and then we draw a line from zero pressure up to this point, which is which is the volume of blood that is within the heart when it is completely contracted. That is the inotropic line. And now this makes sense, right? Because if this line is steeper, say it was like this, then that means that when the SV valve closes here, there would be less blood in it. And that means that the heart was able to contract more, contract with more pressure to push more blood out. And that would be an increase in inotropy, and the slope is increased. So that is inotropy. And that's the inotropic line, which also corresponds to elastance. Now, there's another couple important things here. This dot right here can represent how much blood is in the heart when it starts to contract, right? How much blood is in the ventricle when it starts to contract. And we define that as preload. That's kind of the vocabulary name that we give it. Okay, so preload. Now, if you think about this line here, and this line here, this line right here represents the volume of blood, right? What x-axis is volume. The volume of blood that is in the heart at the end of systole. So, in other words, that's end systolic volume. And the amount of blood here is the amount of blood at the end of diastole, or end this diastolic volume. And remember, stroke volume equals end diastolic volume minus end systolic volume. So stroke volume can be represented by measuring the distance between these two lines. And that would be stroke volume. Okay? Now, we can imagine then, and this would make sense, right? If we decrease preload, if we decrease the amount of blood that comes into the heart before the ventricle starts to contract, that means we move preload over this way. So then our new graph would look like this, right? Inotropy stays the same, but we just decrease preload. Well, then stroke volume would decrease as well. And that makes sense, right? If you don't have as much blood going into the heart, into the ventricle, when it contracts, then there won't be as much blood that leaves the ventricle either. So, stroke volume. Great. Now, there's another principle here, and it's called afterload. But afterload is not what you think it is. You might think, oh, preload is the amount of blood in the ventricle before it contracts. Afterload is what it is after. No, that's wrong. Afterload is the resistance that the blood experiences as it leaves the ventricle. So, for example, as the blood leaves the ventricle and goes into the aorta, if the aorta is smaller, that means there's more resistance, right? There's higher blood pressure in the aorta, and so it's harder for the ventricle to push blood into the aorta. So that means that there is higher afterload. Now, on the other hand, if the aorta is bigger, if it's vasodilated, that means it's easier to push blood into it, and so there's less resistance. And that means that's a decrease in afterload. Okay, so afterload is the load on the heart to prevent the blood from leaving. And we can represent afterload by this. It's kind of this entire green line, right? Because this is the ejection. So how much blood ejects and how much pressure is required to eject that blood is a sign of how much resistance there is, and that's a sign of afterload. But we can just use this dot here for simplicity. And things work like that if we think about it this way. Okay, now say we were to increase afterload. Now, if we increased afterload, then it might be right here, right? And then we'd have a new line that goes like this. It wouldn't decrease at all. I'm just drawing this so that we can see it. The only thing changing is afterload, right? then that means something really interesting, actually. Means that because inotropy is staying the same, then we we have to follow this inotropic line to, um, for this point in the graph. So whenever anything moves, it has to still follow the slope of the inotropic line, because assuming inotropy stays the same, right? Assuming that the heart contracts at the same, like the inotropy stays the same, the contractility of the heart is the same, then that means that if you increase afterload, then the end systolic volume also decreases. And I hope that, I mean, that makes sense logically, right? If afterload increases, if there's more resistance to get the blood out of the heart, then you can't push as much blood out. And so the end systolic volume within the ventricle is higher. And that means that there is a lower stroke volume than before. That is afterload. Great. Now, in order to kind of control how, in order to control blood pressure, right? Because blood pressure is very, very important in the body. And then we'll see a little bit later why it is so important. But we're going to talk about how the body can control blood pressure. And this is one way that it does this is through the baroreceptor reflex. So if we have blood right here in the ventricle, and then it comes out, gets pushed into the aorta. If there's a higher blood pressure here in the aorta, then that means that the aorta will be stretching more, right? There's going to be more pressure pushing the aorta out. The aorta will stretch more. And then furthermore, these arteries that come off will also stretch more. And this little bulge, these bulges here are called the carotid sinus. And within the carotid sinus, there are stretch receptors called baroreceptors. And what those do is they respond to stretch and then they send the information to the brain, and then the brain takes an action. So if these experience stretch, then the brain knows, okay, that's an increase in blood pressure. Now, mean arterial pressure or blood pressure, same thing, right? Is cardiac output times peripheral resistance. The way that I like to remember this is you have a map and it leads you to copper, right? So map copper, blood pressure. Um, so cardiac output then is determined by heart rate times stroke volume, because cardiac output is measured in milliliters per minute. To get those units, you have to do the heart rate, which is measured in beats per minute, and then the volume, which is measured in milliliters. So cardiac output is heart rate times stroke volume. And peripheral resistance is proportional, which means you really don't have to know what it's multiplied by. You just need to know that this changes it, right? It's proportional by the length times the volume over the radius to the fourth. And this to the fourth is really important. That means that the biggest impact on peripheral resistance, and therefore the biggest impact on blood pressure, is the radius of the blood vessels. So from this, we can learn a couple of things. The heart can change the heart rate or the radius of the blood vessels, which is vasoconstriction and vasodilation, in order to impact blood pressure. So if the baroreceptors experience stretch and blood pressure goes up, then in order to compensate for that, it wants to bring the blood pressure back down, so it will decrease heart rate. And in order to further bring the blood pressure down, it might also increase the radius of the blood vessels, which will decrease peripheral resistance. And that would be through vasodilation. So if blood pressure is too high, the baroreceptors say, hold up, and the brain decreases the heart rate and increases vasodilation of the blood vessels. And so what this means is that if there is an increased preload, right, increased preload will mean that there's more blood going into the heart when it contracts. That will mean that stroke volume increases, which means that the blood pressure in the aorta will be higher. And then those baroreceptors will sense that and will then tell the body to decrease heart rate. Okay. So for an example, increased preload would lead to decreased heart rate. Great. That's the baroreceptor reflex. Now, once the blood gets past the aorta and then it starts going down into the rest of our body, it will enter into capillaries. And the capillaries are basically the smallest blood vessels ever, and they're what go in between cells. So these little black squares, those are cells with the nucleuses in the middle, not in the middle, but within the cell. And we're going to talk about how these cells can get oxygen and get nutrients that they need from the blood. So there's a couple pressures driving blood in and out of the capillaries. The first is obviously blood pressure, right? So blood pressure is coming in, and the effect that blood pressure has on the capillary, we use a different name for it, and we call it hydrostatic pressure, for HP, okay? So that's the same word for, different word for the same thing. Now, blood pressure starts at like, normal is 120 millimeters of mercury inside the aorta, right? And so way up here in the body, it's 120. But then as blood meets resistance, right, as it goes throughout the rest of the body, it decreases. And ultimately, there's these precapillary sphincters that are some smooth muscle lining lining the end of the arterioles that go into the capillaries. And these will contract according to the blood pressure. So they're smooth muscle, right? So they have mechanical receptors. And so when they stretch, it causes them to contract. And it kind of keeps the hydrostatic pressure at around 30 millimeters of mercury. So it tries to keep it consistent. And this blood pressure, right, that's the pressure coming into the capillary. And so that would mean that it would want to push blood out. Okay. So there's 30 millimeters of mercury pushing blood out. Then a second variable or second pressure gradient that we have here is BCOP. And this is caused by a bunch of big proteins that are in the blood, primarily albumin. And now these proteins are non-permeable to the capillary walls, so they cannot cross the capillary walls. And this means that they can contribute to osmotic pressure. And that's what BCOP stands for. It stands for blood colloid osmotic pressure. Colloid means a big protein. That's what colloid means. So blood colloid osmotic pressure means the osmotic pressure caused by colloids or big proteins in the blood. Now, if you were to imagine that there's only water out here, then and there's a bunch of albumin inside the blood, that'll cause blood to come in, or sorry, fluid, just in general, fluid in general to come in. And so that pressure is approximately 28 millimeters of mercury. Okay. Now, there isn't just water in the extracellular matrix, right? And so there are some proteins here, and they are also non-permeable. Okay. And we call those ICOP. Or we call the pressure exerted by these proteins ICOP. And that is approximately 8 millimeters of mercury. And so that pressure, that osmotic pressure, right? That interstitial colloid osmotic pressure, so big proteins in the interstitial fluid that cause osmotic pressure, is going to cause blood or fluid to come out, right? And the pressure that will cause that is 8 millimeters of mercury. Great. Now there's one last pressure to consider that affects capillary exchange, right? This is capillary exchange, is IFP, and that's interstitial fluid pressure. And that is caused by these lymph vesicles or lymph vessels. And so what happens is the lymph vessels, they have little valves here. And when you move your muscles, right, just like veins, when you move your muscles, that pushes fluid up because of your muscles contracting. And then these valves will stop it from going back. And so it basically creates a sort of vacuum here. And that vacuum kind of is just continually sucking fluid into it. And that pressure of the sucking is 3 millimeters of mercury. Now, overall, then, what we see here is in the artery, in the arterial end, you have 30 millimeters, 30 milliliters of mercury of pressure going out, 28 coming in because of BCOP, 8 going out because of ICOP, and then a further 3 going out because of the lymph vessels, because of IFP. And this results in a net flow out, right? Because you can kind of see that 30 plus 8 plus 3, right, would be 41. 41 minus 28 is 13. So it's a net 13 millimeters of mercury causing blood to come out on the arterial end. Now, what happens is as blood continues to be pushed down the capillary, right, it eventually gets deoxygenated because of the cells, they're pulling oxygen from the blood, and oxygen diffuses freely across the membrane, right? What happens is hydrostatic pressure, or the pressure exerted by blood pressure, right? The blood pressure actually decreases. And so over here on the venous end, it's actually only around 10 millimeters of mercury. Now, everything else stays the same, but blood pressure is decreased. And so that means that we lost 20 millimeters of mercury. And that means that this 13 millimeters of mercury will go down by 20, which would be -7, right? So -7 millimeters of mercury of pressure leaving. And that means that there's 7 millimeters of mercury of pressure causing fluid to come into the capillaries. And that allows for fluid to come back into the blood, and then it kind of pushes it through the veins a little bit. And then the muscles contract and continue to push the blood. Okay? So that is how this all works, and this is how the pressures kind of differ. And if any of these pressures change, it can potentially cause either dehydration of the cells or edema. And edema is build up of fluid in the interstitial matrix, in the interstitial fluid. Okay, so too much fluid is leaving the capillaries. This can be caused by a variety of reasons. I'll talk about hydrostatic anemia. And hydrostatic anemia, right, relates to hydrostatic pressure. And what happens is these precapillary sphincters are decently strong, but when mean arterial pressure gets above 160 milliliters of mercury, then the precapillary sphincters can't really handle this. Now, mean arterial pressure is not systolic pressure. Mean arterial pressure is the average blood pressure between systole and diastole. And if that gets above 160, which is really high, right? That's really high. What happens is these precapillary sphincters get overrun. They're not strong enough to balance it out. And so the hydrostatic pressure changes from 30 to maybe 50, right? It goes up. And that means that the pressure that's causing fluid to leave the capillaries also goes up. That means that more pressure will leave. And further over here on the venous end, that means that the hydrostatic pressure will also be higher here, which means there's going to be less fluid coming in. So overall, there's going to be a bunch of fluid accumulating in the interstitial space, and that will cause, well, that is the definition of edema.